So hello everybody and welcome back to my YouTube channel and आज के वीडियो में we will be discussing about the CSIR examination. So along with CSIR, IIT JAM, GAT, DBT, all of these examinations that are going to be very important for postgraduate students, right, for pursuing their higher careers. And there are certain topics or units that are there in CSIR, right, or any examination. So there are systematically divided, and each unit has its own importance and all important topics or FAQs, right? So like there are this particular topics: molecular interaction relevance to biology, cellular biology, molecular biology, fundamental process, cell communication signaling, developmental biology, system physiology, inheritance biology, diversity, ecology, evolution, applied biology, methods in biology. So we all come from different backgrounds. Somebody comes from BSc, somebody comes from B Farm, somebody comes from some other allied fields, right? So there is something or the other that we lack, that we don't miss. Like from pharmacy background, we don't study much about evolution, ecological forms, diversity, and all. But those who are coming from other backgrounds, they would have been studying this, right? So there is something or the other that some or the other class would miss. Now how do we cope up and cover it up in other units is important. So today you'll be discussing about unit wise. So the first and a very important is the first unit, molecular and their interactions. So you need to know the atom. So atom, who discovered atom, the different scientists who are associated with the discovery of atoms, and so that is very important. Molecules and chemical bonds. So you need to know about sigma bond, pi bond, coordinate bond, dative bond. All these bonds, there are questions: which is a strong bond, which bond is a coaxial overlap. Next come about the composition, structure and function of biomolecules. They can be macromolecules or micromolecules. They can be vitamins, enzymes, lipids, proteins, amino acid, nucleic acid, carbohydrates. So you need to know the classification like carbohydrates. So aldose, ketose, homopolysaccharide, heteropolysaccharide, monosaccharide and all. Triose, tetros, lipids. So conjugated lipids, derived lipids, proteins, glycoproteins, lipoproteins, phospholipids, all those things, right? Nucleic acids, so different purines and pyrimidine, the base is involved. Then vitamins, different types of vitamin, water soluble, fat soluble, and the deficiency of the disease, that vitamin function on role. Next comes the stabilization interaction. So in proteins, we have quaternary structures. So if there is different bonding involved, hydrogen bonding, there is lot of other bondings like electrostatic forces of attraction, van der Waals forces, है ना? So there are different bonding interactions that are involved. So pi stacking and all. So you should be aware about the stabilization and like alpha helix, beta pleated, about different bonds. Next up is the principles of biophysical chemistry. So this is basically all your thermodynamic chapters, say colligative properties, then reaction kinetics, first order, zero order, pH, buffer, Henderson-Hasselbalch. There are a lot of numericals that are being asked. The different buffer system, acetate, phosphate, borate. Then bioenergetics. So there are different cycles: glycolysis, Krebs, HMP, shunt, gluconeogenesis, glycogenesis, right? ETS. So you need to know about those transporting systems and those different kinds of cycles in our body that cover, and different inhibitors like rotenone, iodoacetate, fluoroacetate, which is the irreversible steps, in which step there is ATP generation, in which step there is a NADP generation, how many ATP are formed and all. Next up, we have principle of catalysis. So this is basically enzyme kinetics. We have Line-Bow box plot, Michaelis-Menten plot. This way often questions are asked. Then from Ott-Hill, oxidoreductase, transferase, and all of those enzymes. Then next comes the conformation of proteins. This is their favorite plot. So Ramchandran plot. So you should need to know the value of psi in the Ramchandran's plot. That like there are, suppose there are four quadrants. So in which quadrant there is L alpha amino acid, then which quadrant there is D, like, right? So you should be knowing about that plus minus and all. And then it is used for secondary structure of proteins. Then different domains, motifs, conformation of different DNA structures like bDNA, aDNA, zDNA, how many base pairs, which is right-handed coiling. Then you need to know about different kinds of RNA: siRNA, small interfering RNA, miRNA, microRNA, snRNA, soluble RNA, hnRNA, heterogeneous RNA, tRNA, mRNA, rRNA, right? So piRNA, picoRNA, and all. There are so many different RNA techniques that are currently being used now, right? So, because silencing RNA is widely used in cancer, epigenetic studies, and all different studies, right? Next up, we have stability of proteins and nucleic acids. So, there are different stabilizers and all involved for proteins that you should be knowing. Next is metabolism. So, there are different cycles: fatty acid oxidation cycle, carnitine shuttle, and all. For amino acid, we have transamination, deamination cycles. Carbohydrates, as I mentioned previously, we have glycolysis, and the opposite of gluconeogenesis, glycogenolysis and all, right? So all those things are being asked. Like precursors, which are the precursor for which endogenous substance, like heme ke liye glycine is a precursor, for tyrosine is a precursor for lot of other substances. Tryptophan is a precursor for serotonin, right? So tyrosine and all, phenylalanine for catecholamines. Cellular organization. So cellular organization basically sees with operon. Operon can be lac operon or it can be tryptophan operon. So you need to know which is inducible, which is repressible, which is anabolic, which is catabolic. In the presence of lactose, what will happen? What is IPTG? The medium for in the which there is what is a promoter region, what is operator, what is YZAG. Then you need to know about the families of gene, then the chromatin fibers, so chromomere, centromere, the different forms of chromosomes, metacentric, submetacentric, acrocentric, all those things. Then protein sorting, so there is a technique called FACS, fluorescence associated cell sorting or fluorescence activated cell sorting. Then there is technique called FISH, fluorescence in situ hybridization. So all those things you should know. Then there is FRAP, fluorescence recovery after photo bleaching. So all these are techniques. Then there's DNA microarray, RT-PCR, that they are frequently asked, confocal microscopy, right? So you need to be well versed with this. Then the euchromatin, heterochromatin, which is lightly stained, which is the main DNA part, which is transcriptionally active. Then transposons, the types of transposons, DNA transposons, RNA transposons, the jumping genes. Next up, we have membrane structure and function. So lipid fluid mosaic model, lipid bilayer ka you should be knowing. Protein diffusion, osmosis, ion channels, different types of active transporters, primary active, secondary active, sodium potassium pump, hydrogen sodium potassium 2 chlorine co-transporter, sodium chlorine symporter, all those things you should know, sodium iodide transporter, right? Those systems. Then regulation of different transporters. Then structural organization. So you should be knowing about the cell wall, nucleus, plastids, peroxisomes, vacuoles, lysosomes, the function, which is a cell or suicidal body, which is a mitochondria powerhouse of the cell, ER, SER, RER. Then there is Golgi bodies, what are the functions, cell wall, teichoic acid, N-acetylmuramic acid, GAG, which is glycosaminoglycolysis, NAM, NAG, N-acetylmuramic acid, N-acetylglycosamines. So all those things. Then cell division. So cell division is mainly CDK, cyclin dependent kinase, CDK 1, 2, 3, 4, A, B, C, D, E. It comes at which phase it activates or it's involved in G1, S, G2, M. So mitosis, meiosis, which is reduction, equational, and how many daughter cells are formed, what are the different mitochondrial microtubule, microfilament inhibitors, colchicine and all. So those things are being asked. Regulation steps, very important. Next up, we have microbial physiology, so that growth curve, bacterial growth curve can be a part of this, growth yield, strategies of cell division, and a stress response, that is again important. Then fundamental processes, we have DNA replication, so extra chromosomal transcription, translation, different Okazaki fragments, Shine-Delgado sequence, Kozak sequences, Hogness box, Tata box, Bribnow box, all those things you should be knowing from DNA point of view. Then enzymes, DNA ligase, DNA polymerase, RNA primase, DNA topoisomerases, all these enzyme involvement. The drugs inhibiting the antibiotics acting on these enzymes, peptide transferase and all. Right, so you should be knowing from promotion, initiation, elongation, transcription, EFTU, EFPI, all those factors. Then from caping, splicing, tailing, post-transcriptional PTM, post-transcriptional and post-translational modifications, machinery, transcription machineries that are involved, right? So methylguanosine phosphate, adenylate residues, all these things are you should be aware about. So next up, moving ahead, we have RNA processing, polyadenylation, same thing that we have discussed, protein synthesis. So in this, you have translational inhibitors, you have tRNA synthetase, amino cell tRNA synthetase, DHU arm, middle arm, T epsilon C arm. So there are different arms involved, and genetic code. So Hargobind Khurana, and you should know the different types of properties of genetic code, ambiguity, free from ambiguous, degeneracy, polarity, triplet and commaless, all those things, right? So next up we have gene expression. So gene expression, gene silencing, regulation of expression of FAG, vectors, plasmids, cosmids, yeast artificial chromosome, bacterial artificial chromosomes, all the vectors you should be knowing, right? Viruses, which are the different viruses that are involved, like phage, lenti, sendai, these all vectors, these all can be used as vectors, है ना? Then cell communication. So cell to cell communication, cell to cell junction is very important. So we have a lot of junction, hemidesmosomes, desmosomes, tight junction, gap junction, right? Hemidesmosomes. So they have different proteins, claudin, occludin, cadherin, sinugulin, integrins, selectin, adhesins, lot of molecules that are involved. So you should know the integrity of these barriers, how they act, how are they connected, cell behavior. Then cell signaling said different types of receptors, ligand gated, G protein, RTK, nuclear, G में GS, GI, GO, GQ, wind signaling pathway, beta catenin pathway, right? Wind signaling is widely common pathway, WNT, right? Then you have hedgehog signaling. You should be knowing about hedgehog signaling, orphan receptors, है ना? A voltage gated ion channel, voltage gated ligand channel, ion gated ligand channels. Quorum sensing, so it is involved in biofilm. So like biofilm has a lot of steps, maturation and all. So quorum sensing is one of the part of that. Then we have cellular communication, as I mentioned, the different extracellular matrices, neurotransmitters, neurohormones, neuropeptides, cancer epigenetics, what is the role of TGF, TNF, tumor suppressor gene, tumor growth, transforming growth factor, the different apoptosis, necrosis, the different markers for autophagy, LC, light chain, Becklin, ULK. Then you have apoptosis, bid, bad, BAX, BCL2, pro-apoptotic, anti-apoptotic, caspase, executioner caspase, initiator caspases, proto-oncogenes, oncogenes. Then immunity, innate, inborn, acquired, active, passive, herd immunity, MHC complex 1 and 2. So CD4, which one is for CD8, which is helper, which is killer, which has beta 2 microglobulin factor involved. Then you have a lot of immunoglobulin, IgG, AMED, so the difference in immunoglobulins, the different monoclonal antibodies, so salvage pathway, de novo pathway, furin synthesis, you should be knowing for hybridoma technology. Then PEG medium, HAT medium, right? Different antibodies like omalizumab, daclizumab, trastuzumab, temozumab, bevacizumab, then there are a lot of so many different antibodies that are involved currently in different diseases. Then TLR, so there are different TLR isoform superfamily, TLR 1, 2, 3, 4. So it's like you should know about TLR 4 at least, because it's a lot of inflammation, fibrosis, cirrhosis associated, and that is very important. Then should be knowing about hypersensitivity reactions, type 1, 2, 3, anaphylactic, atopic, retarded, arthritis, cytotoxic, cell mediated, which one has a serum sickness, tuberculin, skin test and all. Then APC, antigen presenting cells, so your beta cells, macrophages, dendritic cells, all these are involved over here. Complement system, C3, C3A, C5, C5A, when is the complement system activated, what are the intrinsic complementary factors, all those things you should be knowing. Moving on, we have developmental biology. So from developmental biology, basically we have mutation, types of mutation. So point, frame shift, cross, spontaneous, induced, addition, deletion, transversion, ring disc fragments, all these types of mutants, mutagenesis. Then there are other types of mutation as well that you should be knowing in detail. We'll be discussing ahead. Right, so cell linkages, so incomplete dominance, complete dominance, incomplete linkage, complete cell linkage and all. You should then imprinting, है that all should be knowing. So there is DNA footprinting and there is DNA fingerprinting. I want you all to type in the comment section the difference between DNA footprinting and fingerprinting. Right, let us know how many people are aware about these two terms. Then gametogenesis, so blastula, gastrula. So blastula maybe have ectoderm, endoderm, mesoderm, different layers formed during blastration, gastrulation. Then zygote formation, then production of gametes, so haploid, diploid, and draw. So there are a lot of different animals as well involved. Like there is involvement of Cyanopeptitis elegans, Drosophila melanogaster, zebrafish, and all these things you should be knowing, right? C. elegans, Drosophila, zebrafish, that are frequently being asked. So just once go through it. Then there is Arabidopsis, Anthurium, floral development basically from apical meristem. So ethylene, all those are hormones, auxin, cytokine, gibberellins, abscisic acid, and one new hormone that you have to study, jasmonic acid. So jasmonic acid is again very important in there. So different vulva formation, study about vulva formation, right? So that is also important in Drosophila and C. elegans, and that is frequently asked. Then vertebrates, invertebrates, embryogenetic, larval formation, metamorphosis, again very important parts. System physiology, maybe have CAM cycle, Kranz anatomy, cycling, light dependent, light independent reaction, photosystem A, photosystem B, cycling, non-cycling reaction, C3, C4 plants, photosynthesis, all those things that are coming. PEP, Rubisco, all the science students would have heard about PEP and RUBP, ribulose biphosphate. citric acid cycle, electron transport, the ones that I mentioned previously, respiratory cycle. So all those things are very important for us to know, right? Nitrogen, we have ammonification, nitrification, denitrification, different bacteria like anabaene, nostoc, rhizobium, and all these bacteria, aerobic, anaerobic, facultative, anaerobic, obligate anaerobic, so that are frequently being asked. Then rhizobium, that is also important bacteria. All these things you should be knowing. Then plant hormones, so I mentioned about the different xylem flow and transport systems, physiological effects of those plant hormones, like which one is involved in fruit ripening, which one is involved in root formation, which one is involved in shoot formation, all those things, because they were asked frequently. Next up, we have sensory photobiology. So, sensory photobiology, we have cryptochromes, we have phototrophins, phytochromes, and then also you need to study about SDP and LDP, which are short day plants, long day plants, photoperiodism basically, morphogenesis, photomorphogenesis, cotomorphogenesis. So, all these things are sometimes as vernalization, bagging, tagging, emasculation, and a stomatal movement, so transpiration and different phenomena related to stomata, right? That is being asked sometimes. So you should be knowing. Next up, we have solute transport and photoassimilation system. So for solute transport means translocation systems, you should be aware. Secondary metabolites may terpenes, phenolic compounds, nitrogenous compounds, alkaloids. Stress biology में biotic and abiotic components. So ecology basically that will be studying ahead. Primary consumer, secondary consumer, carnivores, herbivores. Bregman's rule, Allen's rule, right? So all these rules are very much frequently asked. Next up, you have system physiology. So in this, we have animal system physiology, where we have hematopoiesis, hemoglobin, different techniques for detection of blood components, cardiovascular system. We have cardiac cycle, regulation of cycles, respiratory systems. May we have elimination of waste, regulation, transport of gases, exchange of different gases, like what is the tidal volume, inspirated reserve volume, expirated reserve volume, all those things you should be knowing, right? Then we have nervous system, so neuron action potential, nerve conduction velocity, laser Doppler, very important, that is frequently asked. Then we have a lot of different iPSCs, induced pluripotent stem cells, HSC, hematopoietic stem cells, that are frequently asked in examination. Sense organs, so eyes, nose, ears, teeth are basically very famous sensory organs. Excretory organs, where we have comparative physiology of excretion, kidney, urine, है ना, all those things, GHP, BOP, FHP, blood osmotic pressure, hydrostatic filtration pressure, effective filtration pressure. Then I need to know about certain terminologies. FFS, flipases, flopases, scramblases. This is very oftenly asked. So there are different types of ATPases involved. P type, V type. And ATPases are of different types. V type, P type. So you should be knowing about flipases, flopases, scramblases, clarivate like genes, CLV. They are called clarivate like genes. So they are sometimes asked. So you should be knowing about that as well. अच्छे से. Then different techniques like Edman's reagent, Sagna's reagent for protein sequencing. Another what is phenylazothiosinate and all. Then very important principle, Hardy-Weinberg's principle. You cannot miss this for homozygotes, heterozygotes. Hardy-Weinberg's equilibrium process. So allele frequency and all to calculate, we use this method. Then we have Messels and Stahl's experiment. So Messels and Stahl experiment is also very commonly asked. Hamilton's experiment, Bragman's rule. Then Hamburger's phenomena for chloride shift, है ना, while acid secretion mate is involved. So formation of HCl in our stomach, in our gut. Then Maxim Gilbert sequencing of protein, Maxim Gilbert sequencing. So all these things are very commonly asked in examinations. Moving ahead, we have thermoregulation. So there are a lot of baroreceptors, thermoreceptors, chemoreceptors, mechanoreceptors, tactoreceptors. So what are the different thermoregulatory centers of our body? Then digestive system, may basal metabolic rate, you should be knowing. Then different types of energy calories of carbohydrates, fats, proteins, energy balance, digestion, absorption, the different systems. So basically all the systems that are studying cardiovascular, neurological, nephrological, endocrine, pulmonary, lymphatic, you should be knowing the parts of that system thoroughly, right? Like from all the cells involved, all the glands, and all those things, the functions, Next endocrine में we have neuroendocrine hormones, gametogenesis, spermatogenesis and all. Moving ahead, we have inheritance biology. So we have law of independent assortment, Mendel's law. So you have Mendel's law of segregation and Mendel's law of independence, assortment, dominance, codominance, incomplete dominance, allele, multiple alleles, pseudo alleles, different complementation test, Mendelian principle. Maybe we have sex linkage diseases. So there are AR, XR, AD, XD, autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive. So Huntington's disease, autosomal disease. Then we have a lot of hemophilia, which is X-linked recessive disease. There are a lot of different diseases that are there involved. Cystic fibrosis, ADA, adenosine deaminase deficiency disease. There's a lot of diseases that are Leishman syndrome, you know, that are involved as these. So how do we know the pedigree analysis? Basically, it is called pedigree analysis chart. So you have to study about the pedigree analysis chart अच्छे से. Then we have gene mapping. So what are the different techniques of gene mapping? The HGP, Human Genome Project information. Mitochondria, so mitochondrial inheritance, maternal inheritance, paternal inheritance. Mitochondria, there is a lot of questions that are appearing. Tetrad analysis, pleiotropy. You should know the concept of pleiotropy. So like one gene affecting multiple, or is it redundancy? There is another concept of called redundancy. So study about redundancy as well as pleiotropy both. Then quantitative genetics. So there we have polygenic inheritance, monogenic inheritance, the different heritability, QTL mapping. So QTL mapping is very important. It is associated with quantitative trait locus. QTL is quantitative trait locus. So it is used for a correlation of a quantitative trait in a phenotype. Human genetics. Then we have lethal conditions, incest, somatic cells, germ cells, recombination with homologous and non-homologous transpositions, right? This all is very important. Next up, we have other classifications. So taxonomical classification, genus, order, class, family, species. Then you should be knowing about the different rare, endangered, extinct, vulnerable. The difference between this, like dodo is what, tiger is what, है ना? Species, biotic and abiotic components. So this is all ecology part, है ना? So you should meta population, R and K selection. There are a lot of questions from R and K selection. Then different types of species interaction, community ecology. Then you have ecosystem ecology like CNP cycling, mineral cycling, and terrestrial, aquatic, animals. You should be knowing basics of them. Applied ecology and conservative biology. So you have different biosphere, troposphere, stratosphere, lithosphere. Then about areas, the Paleozoic area, lot of different like those orangutan and all from evolution part. So Charles Darwin theory of evolution, you have to study about that in detail. Then you have Lamarckism, that is Lamarckism is also sometimes Mendelism, Oparin and Haldane effects. Then Avery Macleod McCarthy experiment, right? That is also important. So these named scientist reactions do not leave only gene pool, genetic shift, genetic drift, the difference between genetic shift and genetic drift, kin selection, very important. These are topics that are hot topics. मतलब इससे आते हैं question. Lot of questions pop up from these particular parts. Applied biology में fermentation, upstreaming, downstreaming, transgenic animals, transgenic plants, BT cotton and all, है ना, that are frequently asked. Then different types of endonuclease, rDNA technology. Say you have plus different kinds of vector tools, right? So restriction endonuclease and primer designing and all. उसपे questions are asked. Gene therapy में somatic cell, germ cell, in vivo, ex vivo gene therapy. Then you have a lot of habitat selection, mating, migration. These are few important concepts. Vaccines, diagnostic tests, tissue culture, cell culture, you know, that are very frequent. Culture media. These are McConkey's aggregations, culture media, biosensors. These are very, very important and frequently asked things that come up. Next up, methods in biology. So we have different methods like electrophoresis, gel, capillary paper, so stacking gel, running gel, buffer system in western blotting, southern blotting, northern blotting, lamellae buffer, RIPA buffer, function of those buffers, 2D gel electrophoresis, isoelectric focusing. Then we have a lot of tunnel assay, comet assay, and all this big question are being asked. Then RAPD, RFLP, SSR, single strand breaks. So you have non-homologous end joining, nucleotide excision repair, single stranded break repair, double stranded break repair. These are all DNA repair mechanisms. So and then we have to know about restriction fragment length polymorphism, amplified fragment length polymorphism, all these concepts, RFLP, AFLP, RAPD. ELISA, radioimmunoassay, enzyme linked immunosorbent assay, and all these techniques, immunofluorescence, immunoprecipitation, flow cytometry, all these are very frequently asked. UV visible, NMR, electron spin resonance spectroscopy, X-ray diffraction, so Bragg's law of diffraction and all you should be knowing. NMR, V's index, Miller index, all these things you should be knowing. Coupling constant, McLaughlin rearrangement, Hooke's law, Beer-Lambert's law, Woodward-Fisher rule. Then from statistical method, we have ANOVA, sampling methods, null hypothesis, alternate hypothesis, probability, non-probability, types of error, type 1, type 2, t-test, parametric, non-parametric test, post hoc analysis like Bonferroni, Dunnett square, Turkey's test. Then you have variability, then central tendency, mean, median mode, standard deviation, and all those things are being asked very frequently. Then confidence interval, level of significance, different distribution techniques, binomial, Poisson, normal, and contour plot, regression and correlation, multiple correlations, multiple regression, simple linear regression, multivariate analysis, single component analysis. All these things are being asked very commonly. So you should be well versed. Finally, the last one, radio labeling techniques, radio safety, radioactive compounds, microscopic techniques, STEM, SEM, freeze microscopy, cryo-electron microscopy. So all these things, stereo microscope, patch clamp, electrophysiology, Herg assay, PET scan, MRI, fMRI, CAT, all these things are being asked. PCR, all these techniques you should be know. Pharmacological testings, they are very important from this part. So that's all from this part. I hope that you would have got a lot know about CSI topics and the different subtopics. I'll be making one more video on important concepts of CSI. So please ऐसे ही like करें, share करें, subscribe करें, and also the parts that are involved in this. As a science student, you should be touched upon with life science, cellular biology, molecular biology, environmental biology. These are all the things that are associated with us. So you should be having knowledge about these basics. ठीक है? So I hope this video has helped you. Thank you so much for watching. Do share the video as well with others.